Good morning to one and all present here today. I am Umar. Next person.
Oh, I am Aisha.
I'm Darshini.
I'm Lian.
I'm Rupa.
And I'm Julian.
And we are members of Group 5, and we will be presenting the steps and method of gene cloning. So gene cloning is also referred to as molecular cloning, and it's the process of isolating a sequence of DNA of interest for the purpose of making several copies of it. So let's look at the concept of this process before dwelling into the individual steps. The principle of this process is that a gene is extracted and placed into a suitable vector, which will then produce what is called a recombinant DNA molecule. This vector simply acts as a carrier for this gene of interest and will be transported to a host cell of choice, which is usually a bacterium, but it can be any type of host cell. So let's look at the first step in gene cloning, which is the isolation of DNA. Remember, the term for the specific gene which we want to isolate is called the gene of interest. So an important point to note is that this can be any part of DNA that is desired to be isolated. It can be a promoter or any sequence of DNA. The only important thing is that we match the restricted enzymes, which will be explained in a minute. So the way to do this is by using specific enzymes that we call restriction enzymes. There are two general types of these, either restriction endonuclease enzyme or reverse transcriptase enzyme, which will be included in the PCR reaction, but not in the general one. So there can be several types of restriction enzymes, right? So once the DNA is extracted, it is important to use the same kind of restriction enzyme in the host cell that we are going to extract the plasmid from. This is due to that the process will not be successful because it won't be connecting at the same areas of the DNA. So once the DNA is extracted, it is added with this restriction enzyme. If the enzyme added is restricted endonuclease enzyme, it will create sticky ends where it has acted upon. This means that at the area which is cut, it will create two separate ends which contain the necessary gene. So how it works is that it is able to detect a specific sequence of DNA and only cut at that area. So because of this factor, we need to use the same restriction enzyme later. However, if the enzyme used is a reverse transcriptase enzyme, an RNA template and a short primer for the 3' end, which is complementary to the 3' end, are used by this enzyme to start the synthesis of the first strand of complementary DNA. Now, this template is later used in the polymerase chain reaction or PCR to follow through the gene cloning process, but that will not be explained here. So now the gene is isolated, we can proceed to the next step, which will be explained by my fellow group member.
Okay, so now let me explain the selection of suitable vector and essential characteristic of cloning vector. So before that, a cloning vector means of affecting an organism with an selected strand of DNA or RNA, which the organism can sustainable reproduce by reproducing offspring with the vector. Lian, can you please down the poster?
Darshini.
Or thank you. So based on the selection of the vector, all vector carrier molecules should have few features in general. As example, it must be self-replicating. It must possess a unique restriction site for RE enzyme. Then the introduction of donor DNA fragment must not interfere with replication property of the vector, and they should be easily isolated from host cell. So theoretically, vector characteristic involve three fundamental requirements, which is small size, presence of unique restriction sites, and marker for recombinant selection. So in case you are cloning PCR product, you can always build the restriction site of your choice into the primer and clone in the available vector. So normally, blue-white screening work absolutely fine for cloning, and sequencing, I have found the PKS vector to be very good. So the next one is the characteristic of a cloning vector. So the first one, it must be small in size. Number two, it must be self-replicating inside the host cell. Number three, it must possess some marker genes such that it can be used for later identification of recombinant cell. And the last one, it must possess multiple cloning sites. So that's all from me. So I'm going to pass to the next person.
So I'll be explaining on formation of recombinant DNA and transformation of recombinant vector into a suitable host cell. So rDNA stands for recombinant DNA. DNA is the keeper of all the information needed to recreate an organism. The sequence and number of bases is what creates diversity. DNA does not actually make the organism, it only makes the proteins. By changing the DNA sequence, the way in which the protein is formed changes. Now that we know that DNA, this is where the recombinant comes in. The recombinant DNA is the general name of taking a piece of one DNA and combining it with another strand of DNA. Thus, the name is called as recombinant. So recombinant DNA is also sometimes referred to as chimera. By combining two or more different strands of DNA, scientists are able to create a new strand of DNA. The most common recombinant process involves combining the DNA of two different organisms. How does rDNA works? It works when the host cell expresses protein from the recombinant genes. A significant amount of recombinant protein will not be produced by the host unless expression factors are added. Protein expressions depends on the gene being surrounded by a collection of signals in which it provides instruction for the transcription and translation of the gene by the cell. The signals includes the promoter, the ribosome binding site, and the terminator. Expression vectors in which the foreign DNA is inserted contain the signals. Problems are encountered if its gene contains introns and contains signals which acts as the terminators to be a bacterial host. This results in premature termination, and the recombinant protein may not be processed correctly, folded correctly, or may even degraded. What are the three methods of creating recombinant DNA? Transformation, phage introduction, and non-bacterial transformation. How is recombinant DNA made of? There are three different methods. The first one will be transformation. The insert contains a selectable marker which allows for identification of recombinant molecules. An antibiotic marker is often used to a host cell without a vector dies when exposed to a certain antibiotic, and the host with the vector will live because of its resistance. The vector is inserted into a host cell in which a process called transformation. One example of a possible host cell is E. coli. The host cells must be specially prepared to take up the foreign DNA. Selectable markers can be for antibiotic resistance, color changes, or any other characteristics which can distinguish transformed host from untransformed host. The second one would be non-bacterial transformation. This is a process very similar to transformation, which was described above. The only difference between the two is non-bacterial does not use bacteria such as E. coli for the host. In microinjection, the DNA is injected directly into the nucleus of the cell being transformed. In biolistics, the host cells are bombarded with high-velocity microprojectiles, such as particles of gold or tungsten that has been used to coat the DNA. The third one will be phage introduction. The phage introduction is the process of transfection which is equivalent to transformation, except the phage is used instead of bacteria. In vitro packagings of a vector is used. It is to produce phage plugs which contain recombinants. The recombinants are recreated and identify by differences in the recombinants and non-recombinants using various selection methods. Foreign DNA is inserted into expression vectors. That's all from me. I'll pass it to the next person.
So now I will be explaining the part of isolation of recombinant host cells and multiplication of selected host cells. The transformation process generates a mixed population of transformed and non-transformed host cells. The selection process involves filtering the transformed host cells only. For isolation of recombinant cells from non-recombinant cells, the marked genes of plasmid vector is employed. For example, the PBR322 plasmid vector contains different marker genes such as ampicillin-resistant gene and tetraxylin gene. So in the next step, they will be transformed in the transfer into the Petri plate. The Petri plate contains antibiotic ampicillin. Only cells resistant to the drug can grow, which means that non-transformed cells left of plasmid in the ampicillin will die. The cells with plasmid are resistant to ampicillin and survive. After that, they will be introduced into culture media. So which will be optimum parameters to allow them to grow and multiply. At this stage, the host cells divide and redivide along with the replication of the recombinant DNA carried by them. If the aim is obtaining numerous copy of GI, GL, then simply replication of the host cell is allowed. But for obtaining the product of interest, favorable condition must be provided such that the GL in the vector expresses the product of interest. So moving on is the isolation and purification of the product.
Okay. I will explain the next step, which is the last step, isolation and purification of the product. Okay, this next step involves isolation of the multiplied GI attached with the vector or of the protein encoded by it. This can be rightly compared with the process of harvesting where we collect the crop from the field. Before they do this, usually it will make a screening for clones with desired DNA insert. Modern bacterial cloning vector use the blue-white screening system of distinguished colonies of transgenic cell from those that contain the parent vector. In this vector, foreign DNA is inserted into a sequence that encodes an essential part of beta-galactosidase, an enzyme whose activity resides in formation of a blue-colored colony on the culture medium that is used for this work. Insertion of the foreign DNA into the beta-galactosidase coding sequence disable the function of the enzyme so that colonies containing transformed DNA remain colorless. Therefore, experimentalists are easily able to identify and conduct further studies on multiplied GI colons while ignoring those that do not contain recombinant DNA. This is followed by purification of the isolated gene, copy or protein. Purification is the chemical context in the physical separation of chemical substances of interest from foreign or contaminating substance. Pure result of a successful purification process are termed as isolate. This means that we will isolate all the cloned gene from the chemical substances that be used for them to grow and multiply. So at the end, we will get the gene cloning that we want. That's all from me.
Hi everyone, I will begin discussing on the methods of gene cloning. This describes the process of copying fragments of DNA, which can then be used for many different purposes, such as creating genetically modified crops or finding a cure for a disease. There are two types of gene cloning: in vitro, which is using the polymerase chain reaction (PCR) method to create copies of fragments of DNA. The other type is in vivo, which involves the use of restriction enzymes and ligases using vectors and cloning the fragments into host cells. So the polymerase chain reaction, or the in vitro method for making many copies of a specific section of DNA without the need of for vectors or host cells, the DNA to be copied, also known as the template DNA, is mixed forward and reverse primers complementary to the end of the template DNA. Nucleotides and a version of DNA polymerase known as Taq polymerase, which is the enzyme which is stable under high temperatures and is obtained from the thermophilic bacterium Thermus aquaticus. This whole cycle involves the repetition of three main steps, which are firstly denaturation, which separates the two nucleotide strands of the DNA molecule. The second step is primer annealing, in which the primers bind to the single-stranded DNA. And the third one is extension, in which nucleotides are added to the primers in the 5-3 direction to form a double-stranded copy of the target DNA. Each cycle takes a few minutes, and repeated cycles can produce large amounts of a specific DNA sequence in a matter of hours rather than days. However, this cloning method does require knowledge of some details about the nucleotide sequence to be copied, and the technique is very sensitive to small amounts of contamination. Thank you, that's all from me.
Last but not least, I will be explaining about gene cloning in vivo. So in vivo, which involves the use of restriction enzymes and ligase using vectors and cloning the fragments into host cells. For in vivo cloning, a fragment of DNA containing a single gene or a number of genes is inserted into a vector that can be amplified with another host cell. A vector is a section of DNA that incorporate another DNA fragment without losing the capacity of self-replication, and a vector containing an additional of DNA fragment is known as a hybrid vector. So there are 4 types of vectors, which is plasmid vectors, lambda phage vectors, cosmic, and also expression vectors. So if the fragment of DNA includes one or more than one genes, the process is referred to gene cloning. So what is gene cloning? Gene cloning is the process in which gene of interest is located, a copied clone out of all the DNA extracted from an organism. So the basic steps in gene cloning are: the first step is the DNA extracted from an organism known to have a gene of interest is cut into gene-sized pieces with restriction enzymes. Secondly, the bacterial plasmids are cut with the same restriction enzyme. And thirdly, the gene-sized DNA cut into plasmids are combined into one test tube. Often a plasmid and gene size of DNA will anneal together, forming a recombinant plasmid known as recombinant DNA. Fourthly, the recombinant plasmid are transferred into a bacteria using electroporation or a heat stroke. Last step is the bacteria is plated up and allowed into cloning, and all the clonings are plated into called gene library. So gene library is a screen to identify the clone containing the gene interest by looking for the DNA sequence or a similar gene. The importance of gene cloning is the DNA molecules produced through the cloning techniques are used for many purposes, which are DNA cloning can be used to make proteins such as insulin with biomedical techniques. It's also used to develop recombinant version of the non-functional gene to understand the functioning of the normal gene. This is applied in gene therapies also. Last important is it helps to analyze the effect of mutation on a particular gene. So in summary, what do we learn? We learned that gene cloning starts a new age of modern biotechnology. Gene cloning is also being used to address problems in all areas of agriculture production, pharmacies, environmental issues, food and nutrition, and many other things as well. Classic gene cloning involves the following steps. First is restriction enzymes, dissection, and ligation. Second is isolation of DNA, ligation, transfection, and selection.
Lastly, it involves gel electrophoresis. So that's all from us. Thank you so much.